All right. Now let's finish it. So this pathway, done. Done. And not that hard, but another thing to learn. Just, yeah. Knee bone connected to the leg bone connected to the hip bone or whatever. Just keep going. Run down the connections. Make sure you can do it forward, backward. Yeah. Not sideways. That doesn't make any sense, but make sure you got it. All right. So, specifically, what are we talking about here? Well, phosphorylated ERK. So ERK kinase gets phosphorylated. Then it can go to the nucleus, and then it can transfer that phosphate, or it can phosphorylate ELK1, which is a transcription factor that will make the gene called FOS. It'll express a FOS gene. And FOS is actually a transcription factor. This is one of those FOS and June. These are those oncogenes. Viruses will sometimes bring in copies of these that are a little bit defective, like they're always on, and they'll just release a bunch of them, and all of a sudden the cell will start dividing. That's just one of the things that can happen. In this case, that's not what's happening, but this is going to be part of the process that's going to induce T cells to divide. So we're going to turn this on for a second. Okay, so ERK is going to phosphorylate ELK1, and ELK1 is going to cause expression of FOS. If you're interested, this is the name of the particular DNA sequence that is bound by ELK1. We don't need to know that. And SURF is going to be another thing that binds with ELK1. We don't need to know that. Yeah. Okay. So junk is going to be phosphorylated once again, because why wouldn't it be? And junk is a kinase, and so it's going to come over here, and junk will be able to enter the nucleus, where it's going to activate June by phosphorylating it, which will allow then FOS and June to come together, and FOS and June together make what we call AP1. This is a particular transcription factor made of pieces of two other transcription factors, and this will drive a whole bunch of different things happening. So that's the next thing that happens. So these are the kinases that are going to be set off. So we've got, going back, RAS GTP is like a can opener. It's going to pry that old GDP out of RAS. Open RAS up so that it can now accept a new GTP. And when it does, it's going to phosphorylate RAF, which will phosphorylate MEK, which will phosphorylate ERK. ERK will go to the nucleus. ERK will bind ELK1. There it is right there. And it's going to transcribe a gene. That gene is going to be the FOS gene, because there's already June floating around. Junk will get activated as part of this process, and it is going to come in here and help activate this complex. But the June was already there, so we're going to add a phosphate to it. We're going to join a FOS that we just made to a June, and this is going to be activated via sort of downstream things that happen from some of the other kinases that were activated just upstream. Anyway, that is then going to start transcribing genes. So like here they're showing cyclin D1, and so we're going to drive the cell cycle, basically, round and round and round. And this is one of those topics where I'm pretty sure everybody else in biology would know more about cell cycle than I do, and that's fine. Yeah. That is just fine. So if you want to know more, go ask literally anybody else in the department. Or look it up. That's actually a smart thing to do. Okay, so that's that pathway done. So this is how we can cause the cell to start to divide by, remember all the things that happened before this? Remember that it was PLC gamma that cut the PIP2 in half, and whatever, the head group did its thing and set off various genes there. But now the leftover part, the diacylglycerol, DAG, is the base for RAS GTP, which is the thing that will activate RAS, which will then phosphorylate RAF, then MEK, then ERK, which goes to the nucleus and phosphorylates its good friend ELK1, and ELK1 will then be the transcription factor responsible for recruiting polymerase to transcribe the FOS gene. Yeah. Okay, some of those other kinases, I don't even remember which one, maybe multiple of them, but several steps down line, I think we're going to get junk activated. It's going to be phosphorylated, and that's going to come over here and phosphorylate C-Jun. And C-Phos, the C, means that it's the cellular version, like the normal version. The original versions of FOS and Jun were viral genes. And so this is how we discovered that these are things which can modulate the cell cycle. They were from a virus that actually induces cancer, basically constant cell division, because it had a FOS and it had a Jun of its own. And so it didn't have to wait for all this signaling stuff to happen. The virus just made them. It's like, here, I made these for you. Maybe go divide. Yeah, because viruses do really well in fast dividing cells. That's the idea there. So anyway, they'll bind to, they together form AP1, which binds to an AP1 binding site, because of course it does. And then that will cause transcription of cyclin D1, among other things. AP1 binding site has a particular sequence. You don't have to learn it, but it exists. It's part of the world of things that people know. Yeah. All right. So let's tell some other stories about the old diacylglycerol. Yeah. So after the IP3 got cut off, remember diacylglycerol, wandering alone, like, oh, what should I do? What should I do? Well, here you go. One thing it does is it binds to protein kinase C theta, PKC theta. And that's this thing. The theta is the one that looks like an O with a line through it. PKC theta will actually bind multiple copies of karma-1. And so you get this great big sort of scaffold with karma-1 and MALT1 and BCL10. Karma-1 gets phosphorylated by protein kinase C theta, and that's how this starts. So there's a dot here, but PKC theta put the dot on there, put the phosphate on there. So then these will be able to assemble once they are phosphorylated, which is great. And so once we have a nice little complex of these, it will form a landing spot for TRAF6. Yeah. So think it's like, I don't know, traffic. Traffic builds up and TRAF6 comes along to watch. It's one of those, you know the drivers that always slow down when there's a wreck on the highway to just look and see if they can see some cool stuff. Yeah, TRAF6, totally that type of protein. Yeah, not actually. Okay, TRAF6 is going to bind this thing, which is called IKK, which is IKK. IKK has multiple subunits, an alpha and a beta and a gamma subunit that is also called NEMO, confusingly. This is going to already be assembled with a polyubiquitin tail. So the little orange triangles are going to be ubiquitin. And so what happens here is that TRAF6, once it binds, will be recruited, and it'll make a scaffold for polyubiquitination. And so other proteins, ubiquitin ligases, and there's a whole cell biology lecture on those, but they will come along and they will only attach ubiquitins when TRAF6 is bound to a great big pile of MALT1, BCL10, and PKC theta. Yeah. So what comes along next? Why? It's going to be this complex. It's the TAB-TAC complex. So it's got tab 1 and 2 are these two little dots, and TAC is a kinase part. And so tab 1 and 2 and TAC together can only be active. The kinase is only active when it's bound to a polyubiquitin chain, which is going to be bound to TRAF6, which is bound to the complex, which is bound to Beckel10, MALT1, and Karma1. PKC theta is the thing that put the phosphate on and allowed all this to happen. And it could only do that because it was stuck to the membrane and activated by binding a diacylglycerol, which only exists because, phospholipase, PLC gamma. Phospholipase C gamma cut off a diacylglycerol from a PIP2 molecule, etc., all the way back to the beginning of time, which starts right about with CD45. Dephosphorylating something. Yeah, go back and look it up if you don't remember. So I think our lectures are going to consist of going backwards and forwards through this pathway. And that's what your studying should look like as well. Backwards and forwards, over and over. Yeah, till you get it, till you can just say it, like your address, like your phone number, like your name. Say it enough times and it'll stick in there. So Tab and TAC are now going to be activated, and TAC is going to phosphorylate IKK. And once it is phosphorylated, that will phosphorylate IKB. And IKB is the inhibitor of NF kappa B. So that's I kappa B. Sorry, that's a little k. So phosphorylates I kappa B. And that's going to cause ubiquitin to be added to I kappa B. Once it gets phosphorylated, now it's a target for ubiquitination. And so once that happens, I kappa B is going to be degraded, which means NF kappa B is finally free, to carouse or whatever, to be a transcription factor, to go to the nucleus and do stuff. So running this backwards, once there was a transcription factor called NF kappa B, unhappy home life, because it was stuck to an I kappa B, an inhibitor, specific inhibitor of NF kappa B. Oh my gosh, yeah. I hope you don't end up stuck to an inhibitor at some point in your life. Anyway, these two are paired. You can't take them apart unless somebody else comes along. And so who comes along? But it's TAC1. And TAC1 is going to put just a little tiny phosphate onto I kappa B. And I kappa B is going to get tagged with ubiquitin. And when you get enough ubiquitins in a chain, then boom, you get hauled off to the proteasome and degraded. Yeah. And just dissolved, basically, back into amino acids. And then NF kappa B gets to go into the nucleus and be the transcription factor it could have always been. Like, wow, dark story, I guess. Yeah, but do you remember it? Yeah. Please come up with some better way to remember this, please. But whatever, yeah. Whatever works, we'll be fine. Okay, so Karma, Malt, Beckeltan complex, TRAF6 sits on complex, is ubiquitinated. Tab 1 and 2 come along, they recruit TAC. TAC1 will attack IKK, so it'll activate IKK. So there, it put a little phosphate on. This is TAC1. Boop, phosphorylated. And now IKK will phosphorylate I kappa B. I kappa B, when it's phosphorylated, will now get ubiquitin chains added to it, like ubiquitins stacked on top of each other. And that, my friends, is a signal leading to degradation. So these are ubiquitin chains and they don't lead to degradation. These are ubiquitin chains and they do lead to degradation. There are different ways that you can hook ubiquitins up, and it has a different meaning basically inside the cell. So different numbers of them and different ways that they're linked together can have different meanings. So that's why it looks like it's just one protein, ubiquitin, which it is, and it's getting two very different effects, because we're not getting destruction of IKK, and we're not getting rapid destruction of TRAF6 after it gets ubiquitinated. We're only getting destruction of I kappa B because it has that kind of ubiquitin. I think I'm gonna say the wrong thing. There are a couple different lysines to which you can add the next ubiquitin, and that's part of what determines the function that they will have and how they're hooked together. Yeah, that's the idea. Okay, fine. Wow, that was a lot. Yeah, only a little bit left to go. But let's pause here and we'll do this last section in a third little baby part. So that's fine. You can go get a nice little cup of tea, nice little biscuit of some sort, little cookie. And then, meet me back here for the last part's like 5 slides, like 5 minutes. Not too bad. All right, see ya.